Hi, my name is Gustavo Pacheco, and I am an MD-PhD student in the Medical Scientist Training Program at the University of Virginia School of Medicine. As a graduate student in the lab of Douglas DeSimone in the Department of Cell Biology, I seek to understand the mitochondrial and metabolic processes that underlie complex collective cell movements. Specifically, we seek to apply in vivo imaging and metabolic assays to study the association between dynamic cell rearrangements in embryonic gastrulation, as highlighted in this movie, and the metabolic shifts in mitochondrial respiration and glycolysis that occur during embryonic development. We focus our investigations on the migration of mesendoderm tissue during gastrulation, which collectively extends along the fibronectin-coated blastocele roof, as demonstrated in this movie. As diagrammed, this collective migration is organized by a gradient of high traction stress cell extracellular matrix adhesions mediated by alpha 5 beta 1 integrin proteins. Furthermore, there is an opposing gradient of intracellular tension mediated by C-cadherin adhesions. Notably, when we examine the in vivo migration of the mesendoderm in the presence of mitochondrial membrane potential biosensor tetramethylrhodamine ethyl ester, or TMRE, as shown in green in this movie, we found that leader cells produce high TMRE signal with a progressive decrease in follower row cells. This distribution of mitochondrial membrane potential in the tissue correlated with alpha 5 beta 1 integrin traction stress. To test if alpha 5 beta 1 integrin signaling could be regulating the distribution of mitochondrial membrane potential, we examined mitochondrial membrane potential of the mesendoderm in the presence of fibronectin fusion proteins, which induce inactive and active integrin conformational states. Both in vitro and in vivo approaches demonstrate high TMRE signal when integrins were in the activated conformation and low TMRE signal when integrins were in the inactive conformation. Such findings indicate that integrin activation alone can drive mitochondrial membrane potential in the mesendoderm. Taken together, our studies offer new insights into the role of the mechanical environment outside of the cell on the metabolic processes within the cell. Ongoing studies will unveil the specific metabolic processes that are controlled by integrin signaling. Ultimately, we will advance our understanding of the mechanisms in collected cell migration that must be tightly regulated during embryonic morphogenesis and how they can be dysregulated in various pathologies such as cancer metastasis.